Well, thank you very much. It's a real pleasure, and I'm nearly ashamed to admit that it's my first visit to Cornell. It's hard to believe, but true, but better later than never. So today I will talk about the extensive impact of the CIMMYT wheat breeding program, and I had the choice to go into a couple of subjects into great details, or really give you a better overview of what we do. And I have decided to do the second, and I really hope to generate interest. And if you would like to have follow-up discussions, please do. And I'm also would be very, very interested to link you with some of the CIMMYT researchers for those of you who don't have already contacts with CIMMYT. Yep. Okay, what is CIMMYT? CIMMYT is the International Maize and Wheat Improvement Center. It is the initiator of the Green Revolution, and it's today the main supplier of maize and wheat germplasm to a global network of cooperators. It is one of 15 international centers with headquarters in Mexico, and it employs today about 220 scientists and 900 support staff. The 220 scientists come from 50 countries, so we are really multicultural. And our annual budget is now $190 million. CIMMYT has offices basically in all major wheat and maize producing countries in the developing world. And our mission is to sustainably increase the productivity of maize and wheat systems to ensure good global food security and reduce poverty. And I really would like to emphasize that productivity gains is in our mission, and compared to 10-15 years when productivity was even a no-no word for many donors, that has dramatically changed. Then CIMMYT and Cornell, I said it's my first visit, but CIMMYT and Cornell have relations over a long period, and my Director General has just been here recently. Unfortunately, I have not seen some of his slides, and so maybe I go one or two again. Okay, anyhow, we have here, maybe you have seen that one, because I made them, so but I don't know whether he used it. Certainly Ronnie and Mark on breeding and on the durable rust-resistant wheat. Ken Sayer, one of the most knowledgeable agronomists I know. Matthew Reynolds, he is a student of Cornell, but also an adjunct professor. Ravi Singh, and then also Jessica. And our most intensive, our collaboration is at present is in the Borlaug Global Rust Initiative. And since Cornell is leading this, I'm not going to talk much about it. But I have to say that this is really one of the most impressive and efficient cooperations I have seen in my 30 years. This team was able to put together basically all these cooperators. When UG99 broke out, there were basically 5 or 6 people in the world you could consider stem rust resistant. Today we have truly a critical mass of people again, and I think the cooperation and the way Cornell has managed this is really only applaudable. Also the impact, and there we come to CIMMYT. CIMMYT has a little bit to do. These are the varieties which have been released since the outbreak of UG99. And most of these varieties have been released after 2008. Now think about the project started in series about 2006, and since then more than 40 varieties have been released, which are grown on more than 2 million hectares already. I think this is a remarkable achievement, and it was only possible because the research and the policymakers and the donors have worked together. A lot of these varieties are out also because USAID has put in considerable money to support the seed sector. And you can see that the countries which are targeted are really all going from Kenya up to South Asia, Pakistan and India. Okay, now let me explain a little bit about CIMMYT, and some of you may have heard about the shuttle breeding. CIMMYT goes back when Norman Borlaug came in 1943 to Mexico, and his challenge was stem rust as it is today for us. And Norman Borlaug, exactly like plant breeders today, asked the question, how can I accelerate my breeding gains? How can I get faster? How can I do more per year? And what Norman Borlaug started, he started with a shuttle. He was growing the wheat during the summer cycle, from May to October here in central Mexico at 2,600 meters. And for the winter cycle, he moved it to Obregon. And what happens is here, you grow the wheat into longer days which get longer, and here the days get shorter. And Norman Borlaug, I asked him, Norm, did you know that you would develop photoperiod insensitive varieties? And he said, I didn't have the slightest idea about it until we started to test the material around the world. And really what happened was that Norman Borlaug had here developed the photoinsensitive wheat, and this material then was later tested. That's the current approach. It's crossed in Mexico, it goes through the shuttle breeding, and then it's evaluated in Mexico and now also Kenya. And then it goes to all these corroborators here. And I think a big component of the success CIMMYT has is partly this, but more this down here. Because this is where the corroborators around the world share information. They evaluate the material and they provide information back here to CIMMYT. These are about 620 locations. And this, I think, is the backbone of the International Wheat Improvement Network. Without corroborators willing to share the information, the phenotypic information, CIMMYT scientists in Mexico could not make the decision to choose the right parents. And I cannot overemphasize how important this is. In wheat, we still have a public domain-dominated research environment. But once this stops here, I think it will have big impact on the future of international wheat improvement. Okay, another very important component was that Norman Borlaug and today CIMMYT, we send out material around the world. The material very often is tested in environments where it's really not well adapted. But what is done is you select the ones which are a little bit better, and over time you can identify the alleles which increase the adaptation of CIMMYT material. And I will come back to this. I think this is extremely important, and there have been a lot of discussions of separating, regionalizing breeding programs. I think for CIMMYT's wheat program, it's crucial that we continue to develop and test material globally to identify the exception in terms of adaptation, and then we can keep this allele package together. What is also very, very important, and we discussed this, Norman Borlaug and CIMMYT, we did the phenotyping, but today with genotyping becoming important, phenotyping is the bottleneck. And what you see in this slide here, we plan and hopefully will get the fund to establish a global network of phenotyping platforms. And what I mean with this is that we will put then here the infrastructure and also the human resource capacity to really evaluate material for specific traits very well. And that will pay, because everybody today, I mean, sequencing is cheap and fast, but phenotyping is slow and expensive. And if we get some reference locations, and here I have shown the countries and the potential traits, I think that really would help to accelerate our breeding gains on a regional and on a global level tremendously. Okay, now here, this is what I just spoke about. The red countries show countries which have released lines which are derived from the same cross. And wheat is really an exception among all crops, as much as I can say, when it comes to wide adaptation. And I think this makes wheat also in the future a very attractive crop with climate change, because you don't know how the climate will be at a given location tomorrow. And because these cultivars, they are released, some of them are released, one cultivar was released in 40 countries. It has this package of alleles which give you the stability, the yield stability, and also the disease resistance. I come back to this later, which is so important in the future if you have unpredictable weather conditions. For example, if you are a farmer in North Africa and Tunisia, what are you going to grow? A drought tolerant or a variety adapted to high rainfall? The weather can be 200 millimeter or 800 millimeter. And you need cultivars which can respond. And I think this was one of the biggest achievements of CIMMYT, started by Norman Borlaug, to bring these allele combinations together. This is another slide which shows the total adaptation of CIMMYT. Red shows countries which have released selections out of the material from Mexico. And blue shows countries which have used CIMMYT material in their crossing. So basically the material has made a global impact. And here you can see the percentage of lines of CIMMYT which are CIMMYT derived, and as of 2005 We will make over the next 3 years another analysis to look whether this is still the same or whether it's lower or higher. But the biggest impact you can see is here in India. Southeast Asia is 100%, but also Sub-Saharan Africa, most of the varieties which are grown are CIMMYT derived, the same Latin America and also in West Asia and North Africa. On a global scale, around 50% of released varieties are related to CIMMYT. And the impact from this was quite remarkable. In this slide here, you see what had happened if there had been no investments in breeding. And in this case here, the period which was compared, no investments from 65 to 2004, and the impact on the price you can see here on the y-axis. And the highest impact came from wheat improvement in developing countries. And there I think CIMMYT has played a major role. And so I think from this data, they are not from CIMMYT, you can see how big the impact from the international wheat improvement efforts were on food security and in particular price stability. Wheat today in many countries is a crop which doesn't have a very good reputation. Celiac disease, gluten allergy, I mean, you name it, rich man's crop, we can continue. Nevertheless, I'm the director, so I have to sell it that it's a good crop. But in reality, I mean, wheat is the most important source of protein. Many don't know this. Wheat is, after rice, the second most important calorie source, but it plays a crucial role for global food security. And if you look at these countries here, most would associate it with countries which have trouble one way or the other, social unrest. These are the countries with the highest wheat per capita consumption in the world. So if something goes wrong in these countries with the wheat production, many other things will go wrong as well. Okay, and here, this is the last slide on the impact, and then I will go to CIMMYT research, what we do today. But there is a lot of talks about increasing global food production, and that is, many believe there are low-hanging cherries. If you look at this here for wheat, for wheat here, you can see that for any region in the world, this is the adoption of modern varieties. For any region in the world, wheat has the highest percentage. So I could say, great achievements, but what does this mean? If you replace the currently grown varieties with new varieties, you have to replace improved cultivars. And if you replace improved cultivars, yield gains of 50% are just not realistic. Many of the varieties grown here are already very good, so we will have marginal increases in productivity, but the times of the low-hanging cherries are gone. So I think this is really something we have to recognize for wheat and then followed by rice. In other crops, if you replace the varieties, you can get sometimes doubling or tripling of yields. For wheat, this is very unlikely. Okay, and this year what we are all up for, over the coming years due to climate change and other problems, water, diseases, the climate change are clearly reducing the yield. And this here is the forecast what we need over the next 40 years, the yield gains. This is where we currently are, and this is what could happen if we don't start to invest more in agricultural research, in agronomy research, in breeding, in pathology. I think this is really quite striking, and of course, this deficiency, this gap here wouldn't hurt the food production in the US, but it really will hurt people in developing countries. And just to remind you, there are more people living inside this circle than outside of it. For CIMMYT's wheat program, we have basically 3 regions where we really focus on. One is South Asia, a big producer, then North Africa, and West Asia, and then here Central Asia with Ethiopia and Kenya, and the 4th one where we have a very intensive cooperation with is China, but China has a very strong breeding program. And this here, I have to come back here. In this part of the world, this is where global climate change is most likely hitting hardest. And the population growth over the next 40 years in this part here equals the combined population of the United States and Brazil today. So we have to feed already 1.5 billion, and then we have to add another 700 million. And that is really challenging. So one of the projects, which for me are one of the most interesting projects to address what is happening there, is a Feed the Future project, which is led by Kansas State University with Cornell and CIMMYT. And this project is, in my opinion or my knowledge, currently the biggest project on genomic selection going on in the public sector. It will advance the productivity frontier for wheat in South Asia, and it's the challenge of increasing temperature. And what is done in this project is we take the elite lines of CIMMYT, the entire elite lines of CIMMYT, and we test them also in India and Pakistan to get the phenotypic data. We phenotype this material in Mexico and in South Asia, genotype the material, apply then genomic selection, and then hopefully come up with better lines faster. This is for me one of the most exciting projects which currently goes on in wheat improvement, and I'm really looking forward to see the results in 4 years or 5 years. Okay, and this here is some of the models which are done. These are also data from a project CIMMYT is carrying out with Cornell on genomic selection. And what was done here was also that the interaction was considered. And I have to say, for wheat in CIMMYT, we are in a very lucky position, because CIMMYT has pedigrees which go back to the origin, to the landraces. And simply using the coefficient of parentage, in most trials or analysis we have done at CIMMYT, we get already a prediction of about 0.5. And you can see this here when the pedigree comes in this case here, you get very, very good predictions. The markers, when the markers were added, or also the interaction, it increased it to some extent. But the point is, if we combine here the phenotype, the genotype, coefficient of parentage, with the environment and the genetics, the markers, we can come here in all models, in this case, to predictions of about above 0.6. And I think that is already very exciting. And we will see how, well, we will see now how this will help to accelerate. But there is a lot of excitement around this. Okay, now more general. I just said, okay, this method here will tell you on how to make the genetic gains. But there are many other interventions. It will not only be this here. There will be many other interventions which need to be all strong. And as I said before, for me, I personally still believe that germplasm and knowledge sharing will continue to contribute 50%. In particular, in a crop where the research investments are not very high compared to other crops, it's really important that information in germplasm is shared. Otherwise, everybody has to invent the same thing, and that is expensive. The international testing is paramount. Identify the exception. Conventional breeding, we continue to make very good progress. And in Obregon, in Mexico, we were down at about 0.7% per year. And over the last years, also thanks to more expanded funding, we have basically we have 4 times more lines in testing than we had 6 years ago. And that is truly reflected in faster gains. So, I mean, breeding, no matter what, is still a number game. And if you can afford to test a lot of lines for yield, I think that is still for some time to come a good recipe for making progress. Then also at CIMMYT, we have a very intensive collaboration between physiology program and the breeders. And the physiologists, for example, characterize all the parents for physiological traits. Of course, they are also analyzed with molecular data. And then the phenotyping platforms. Currently, we have 2, one in Obregon, which is great, and we have another one in Kenya, which allows to really screen the material for stem rust. But I have shown you that I hope over the next 5 years to add at least 10 more phenotyping platforms, and that really should change the way how the genotype and the genotypic and phenotypic data can be used. Genetic resources, CIMMYT sits on about 160,000 accessions. I come back to it, but only a percentage of it is used. CIMMYT has the biggest collection of translocations in the world, mostly in Chinese spring. We need to invest in the future more in exploiting translocations. Some of the major contributions from 1B, 1R, 1R, 1A, 1R made real boosts on disease and yield. And I think we have the potential to do it, but currently nobody is funding it. Genomic selection, I don't think I have to say this is important, in particular here at Cornell, the Center for Genomic Selection. Synthetics, I come back also on this one here because they have made tremendous contributions to wheat improvement. Physiology, CIMMYT is working on hybrid wheat. We have a research agreement with Syngenta, so we are jointly working on conventional hybrids. CIMMYT is also interested in working on using other transgenic options. There are various out, and so CIMMYT is not anti-transgenic. We are very open to transgenics. We have just received a new biosafety lab. And then finally, what I just said, transgenic and cisgenic. We run freeland trials in Mexico. The government of Mexico is very supportive to give us support for transgenic research. And so we are, I think, a very good partner in this area. Now, wheat improvement at CIMMYT. These here are the traits. And on the left side, you see the traits which every line must have. They are core traits, and they are all tested for this material. So that is yield, of course, rust resistance, drought, heat tolerance, and appropriate end quality. And on the right side, you see here traits which are, depending on the environment, are added. But for example, Fusarium headscap becomes more and more important, and also Septoria. Spot blotch is the disease for the southeast. And what is an emerging disease is 10 spot because of the move to conservation agriculture. Now, one of the things which I really find as possibly among the major contributions of CIMMYT is the development of adult plant resistant material. Adult plant resistant means that in the seedling stage you get an infection, but the adult plant is resistant. And this approach started about 40 years ago. And it was done to get out of this bust and boom cycle where resistance would break down. Here you see various diseases, and what has initially, these genes were introgressed for very specific diseases, for leaf rust, stem rust. But what came out over the last 10 years or so, that these genes really operate differently, and they do not only control one disease, but they control several diseases. And here are some of the genes which have been also some of them cloned. And you can see, for example, that LR34 also is identical with YR18, SR57, PM38, lead tip necrosis 1, spot blotch 1, and BYDV. Now that means if you have this gene here, it will not provide immunity, but it will control all these diseases to some extent. Combine them with some of the other pleiotropic genes, and you really could come to near immunity. And I think that is really important in the future to have such genes for two reasons. One is environments are changing. We see diseases occurring today in environments where they have not occurred in the past. If you have such genes in the background, you have at least protection to avoid disaster. And the other area where I predict that these genes become very popular is if organic wheat breeding becomes more important, and that may be the case in Europe. If you are not allowed to use any fungicides in the future, then you need basically such genes. Fortunately, there are many more coming. I think this is an extremely interesting field of research. And you can see here how much the QTL explains here for leaf rust, stripe rust, and powdery mildew in the range from 4 to 10%. I think that is for QTLs in this area quite remarkable. And what we hope, of course, in the future, and we work with CSIRO, also with the UK, to form cassettes. Currently, if you have these genes, they segregate, and it's very difficult to keep them together. And very few people are really skilled in doing this. I mean, at CIMMYT we have some, but it's really difficult. So if you put them into a cassette, they would not segregate anymore. The second option is that we really come to better markers, reliable markers, because then also it would be possible to keep them together. but one or the other we need in the near future. Now, agriculture and water, and again, I think one of the first publications on the use of water came out of Cornell from Professor Pimentel many years ago. And just to ask the students here, how much liter water do you need to produce 1 kilo wheat? Yeah. I always ask this. So some guesstimates, how much water you need? 1 kilo wheat is about this much. How much water you need? 3 liters? Pardon?
500 liters.
500 liters. That's a lot. That's half a ton for 1 kilo. Other guesstimates? No? 20 liters? Okay, on a global average, it's about 1 ton, 900 liters. And wheat is among the most water efficient crops. For rice, you need 1500, for maize, 1800. 500 liter is the optimum, the absolute optimum, using drip irrigation with the best agronomy. There you can come to 450, but that is not widespread. And the other big challenge which wheat producers in particular will face in the future is that the impact from getting warmer. And why has this such a big impact on wheat? Wheat is the low value crop in crop rotations. And that means the other crops, rice or cotton, their growing period gets longer and longer. Wheat growing is getting later and later, and that means you come more and more into the heat stress as flowering and grain fill. And the big challenge for wheat breeders in these parts of the world, in the developing world, will be to develop cultivars with a faster grain fill period and more tolerance to heat. And this is just, sorry, okay. And here you can see the impact from late planting. This is from the Indo-Gangetic Plains, about 10 million hectares. And you can see if you plant at 5th of November, you can get up to 4.5 tons. If you plant in December, you get basically, well, 1 ton. So this needs to be addressed, and there has been true progress made over the last years. And again, why? Because there was a donor who really funded us well to expand the testing tremendously. The donor was Gates Foundation and USAID. And this allowed us to really test many, many more lines in these environments. And you can see here, this here is the average of the check, and this here is the progress over year. In the last trial now, more than 50% of the entries are above the check, and they are earlier than the check. So the progress in this area, again, is possible, but you really need to combine large-scale testing and using the right markers. This here is an example from our physiology program, where the physiologists now really make recommendations to the breeders, and they use this information, and you can see that from this physiology-derived germplasm, 70% of the new lines have higher yields. So physiology is now also making a contribution to breeding. I mean, physiology, I have to be careful how I express myself. Physiology always made a good contribution, but the linkage and the impact in breeding programs has been so far very often rather limited, and that is now really going to change. Another issue related to water was, and that was a big question to CIMMYT, where old land races had their better water use efficiency than new cultivars, because many of the new cultivars were irrigated. So over 2 years, we had a very, very intensive trial in Mexico where the roots were measured, the water applied, the irrigation, everything was measured. And what you can see here is, this here is the time of release, and this here is the water use efficiency, water used in the plant. And you can see there was no change. However, what changed here is the water uptake. So modern varieties have a better root system to take up more water, but once the water is in the plant, the metabolism has really not been more efficient. So that is also something to look in the future. Now to CIMMYT, I mentioned before, has about 160,000 accessions, and we started to phenotype all these accessions and at the same time genotype. So over the next 3-4 years, we would hopefully complete this, and this information would then be available from the net. Another area where CIMMYT has tremendously invested, and we had some discussions this morning, is in synthetic. That research originally started at Kansas State University in the late 70s, went to CIMMYT, and today, 30 years later, the varieties which were developed and derived from this are released. The synthetics, which are a cross between durum and Aegilops tauschii, have brought new variation on the D genome. And for the non-wheat people in the room, about 10,000 years ago, bread wheat came into existence. It didn't exist before, and that is when Mother Nature made this, well, basically transgenic wheat. That's basically what it is. Right, yeah. Okay, and anyhow, you can see here that we got new resistance for Fusarium headscap, Septoria, spot blotch, rusts, Hessian fly, Russian wheat aphid, and drought tolerance. And we are going to exploit this much more. We are now looking also, these were improved durums using Dicoccum and Dicocoides. The synthetics also showed an interesting trait, like others, that they really absorb more water from the root, from the lower profile. This here shows you the residual water. The green ones are synthetic, so they have a better root system here at antithesis and at maturity than our improved non-synthetic cultivars. And that is the reason, please remember the last slide, they can take up more water, and that's why they are more drought tolerant. It's not really more drought tolerant. And the other thing is also that the synthetics have definitely a much better heat tolerance. As you can see here, this is the check, and here you have the synthetic derived material. So we are really exploiting this now on a very large scale in our crossing program. Yield potential. Yield potential in wheat, there are a lot of discussions. I think if we have a hard look in breeders' plots, we really don't see a plateauing. But we see it in many environments, and one of the reasons is also that in very high yielding environments, a yield, well, may face a plateau. And one of the reasons is that wheat is extremely inefficient when it comes to photosynthesis. So we have formed a wheat yield network, and that will be launched in Obregon in 8 weeks, where about 30 partners around the world are participating. And the areas we are going to focus on is first to improve the photosynthesis. And I will show some results from this one here. Because wheat is so incredibly inefficient compared to other crops that if we could only increase it by 50%, it still would be inefficient, but 50% better. We have to have extra biomass, and that also relates to the spike fertility. And finally, we will use the molecule, transfer these traits into elite material. Okay, I skip this here. Then we have spike photosynthesis. This was really a trait in wheat not researched very well. And how little it was researched, I really only became aware after my physiologist told me. It was recognized 50 years. There were some publications about, but breeding programs have made no effort to use it. Now it's very difficult to measure, so I understand. But if you look at this here, based on the measures in Obregon, the spike contributes about 21 to 41% of the photosynthesis. The flag leaf, another on average 28%, and the bottom leaves really very little. So there is now really a lot of research going on in this here, and we also are in the process here of developing, we found some QTLs, and the genetic variation for this trait in our elite lines was 60%. So I think this is a trait, and we have developed also now the tools to measure it, because it was very difficult, but we can now screen relatively high number for this trait, and let's see where we get in a few years. Another area which I'm really interested is to use airborne remote sensing. And the reason I'm so interested is because the airborne remote sensing already provides better results for many traits than for the ground measure. And in particular, these here are elite lines. And my dream is that over the next few years, we really can come to a correlation between the remote sensing and the yield data. So you can run 20, 30,000 plots and you don't harvest anymore, because you can measure the yield with a reasonable good precision. And then you could run many locations and you wouldn't have the costs of harvesting. Micronutrient deficiency, I have to bring it in because that's really what I worked for many years in Turkey. This is when you apply in Turkey on some soils, but the real issue is micronutrient deficiency in humans. On a global scale, about 2 billion are deficient for iron and zinc. And if you look at iron and zinc deficiency is really a global problem. with the exception of Western Europe and North America. But in particular here in Central Africa and here in the subcontinent, you have a tremendous zinc deficiency. And zinc deficiency has zinc, well, I mean here zinc finger. Zinc is crucial for gene expression, and if you have a mineral which reduces the zinc expression, affects it, then you have a lot of problem. So there are many diseases reported related to zinc deficiency, growth retardation, slow development, increased percentage of cancer. When I was in Turkey, they had made mass scanning, and about 20% of the adults had open spine. You couldn't see it. It was covered by skin, but in X-ray you could see it, and that was related to zinc deficiency of the populations after the Second World War. Now, why do we focus on this part here to develop cultivars here? The reason for this is that zinc is mainly concentrated in the outer layers of the grain, and if you make normal bread, you remove the bran, and then you remove all the zinc. So we needed an area where the whole grain is consumed, and that is here the case, and the other target area where we want potentially start talking with Harvest Plus is in Ethiopia. And we talk a lot about the genotyping revolution in terms of higher throughput, but the progress we made on zinc was only possible because there was a revolution in developing high throughput methods. When the whole started in 2005, you could analyze about 15, 20 samples for about $50 a day. And then came this XRF, and that allows you now for less than, for about 10 cents to analyze thousands. Again, back now we had a tool to screen thousands of accessions, and that was the key for the progress. And that also allowed us to screen many, many more entries, and populations were developed. And now we also have some QTLs, which I always thought we would find, but eventually we were very successful. And in particular, this one here on 2B is extremely important because it enhances the zinc content in the grain and also 1,000 kernel weight. And this is really important because normally if you have shriveled grain, your zinc concentration goes up because it's in the surface. So I think we are really here on something very, very important in order to increase the zinc content in the wheat grains further. With this one, I wanted to stop, and the last thing I wanted to say was, I hope I have generated a little bit of interest of what CIMMYT is doing. We do much, much more, and I also hope that some of you have the opportunity to come to Mexico or to Obregon, where our principal breeding station is, and see what is going on there. I also wanted to say that one of the important components of CIMMYT, aside germplasm, was training. And it was really at the heart of Dr. Borlaug that training of scientists from developing country was absolutely essential to really make the Green Revolution important. We still have a very important training program, and I also hope that I have generated some interest among the future scientists, among the postdocs. I think working at a CG center can be extremely interesting. It can also be challenging, but I really hope that one or the other of you is interested and will contact me. Definitely follow up on the website because the challenges which we face in agriculture are, in my opinion, some of the biggest ones. And I always say flying to the moon was an engineering problem because you could use your mathematical formulas and you could calculate, and if that was okay, you made it. Biological systems are much more complicated. And the challenges we face from water and heat, from less nutrient, we have to produce more with less. I think that is really one of the biggest challenges we will face in research. And I'm sure that sooner or later also the funds will come back much, much bigger than they currently are. Thank you. With this one, I would like to stop. And in spite of, celiac disease and gluten, still wheat we eat. Thank you.